Okay, does that work?
Yes.
Sorry, get rid of this panel. Okay, so thank you very much. I'm really, really excited to be here. And the eagle-eyed among you may have realized that this green thing on the screen is not a fly, but the even more eagle-eyed among you will have realized that there are some fly eggs on this caterpillar. And some of you from the audience, I'm sure, assisted me about a year ago on a Facebook Dipterists group and via Twitter to suggest that I should let this caterpillar drill down into the dirt and hatch out some of these flies. And actually, I think, I've been working on flies for 20 years and probably have reared millions of Drosophila and Anopheles mosquitoes. And this is the first time I really felt truly, I think, maternal towards a fly. About 11 of these tachinids hatched out 7 months later in a little jar that I kept outside, and that it was really exciting. And these exact flies are going to go in for this Darwin Tree of Life project. So I sent a couple over to Chris Raper, who told me what species they were, and a few into the Darwin Tree of Life project, and the rest were released. So I'm working, along with many other people on the project, to try to figure out how we can get around that requirement for dry ice that Lyndall just mentioned. So somebody asked about the pin specimens and whether you can get enough DNA out of pin specimens. And the fact is we can actually get plenty of DNA out of pin specimens and we can sequence their genomes, but the DNA that comes out is extremely fragmented. So it's very hard to put all the pieces back together. It's like if you imagine a jigsaw puzzle, and instead of it being in kind of 10 giant pieces, really easy to put together, it's in millions of pieces and they all look the same and it's really hard to get it all back. So I have connected with Andrew Lucas, who's a hoverfly expert, and he collected all these species this summer. And I emailed him yesterday to say, basically, can you send me a picture of where the hoverflies are right now? And you can see that they're sitting in his home freezer still next to some peas and ice cream. And what we are trying to do is collect these into different preservatives without access to dry ice, and then just normal home freezer to assess whether we can still get these very big pieces of DNA out at the end. The reason these are still sitting in his freezer, though, is because we are now challenged with making sure that we have all of the records of whether he really has permission to collect in the places he's collected. So as part of this project, if you get involved, that is an important thing to keep in mind, is for some of these collections, we need to be really, really rigorous about ensuring we have documentation that we were permitted to collect where these were collected. So I'm going to take you through the journey of a single fly through this system, and this is a ferruginous bee grabber that Liam Crowley collected at Wyden Woods this summer. And this is the actual fly. He collected it, identified it, and popped it in this exact tube, took a picture of it, and you can see this kind of steamy effect, and that's because they're sitting on dry ice to keep that DNA in extremely good condition. So we received that tube. We received hundreds of tubes at Sanger, which is where I work, and it's well known for the Human Genome Project, but it's actually now really invested in this biodiversity sequencing. And we extract the DNA using basically lab kits, and then we evaluate the quality of the DNA using this machine here called the femtopulse. And if you can see my mouse, this is the computer connected to this machine. You put in a plate of DNA into this machine. It takes extremely small amounts of DNA and runs it on a capillary, on a gel, and then reports back what size pieces of DNA do you have in this extraction. And we need these very long pieces. And then somewhat counterintuitively, we actually take the pieces and then we shear them to sort of a similar size, quite big piece still. So we're looking to make them about 20,000 ACs, Ts, and Gs long. For the most important technology we're using at the moment to sequence. And so the DNA then goes into these syringes, and you would be surprised at how much these simple machines cost. This basically machine just moves a syringe up and down at a constant rate and shears the DNA into a very precise size that we need to then sequence it. So once it's sized, we make a library out of it, and then we load that library onto what's called the Sequel2 machine, which looks like this. And then this little kind of drawer opens up, and inside of it is effectively a very high-powered robot that is able to look at the molecules and understand along those molecules by reading them again and again in a circular sort of framework. What are those A's, C's, T's, and G's, and in what order are they, and reports back that information onto a computer. This is what the actual, what's called a smart cell looks like. So it's actually, just postage stamp size, and a little bit of clear colorless liquid is loaded onto that, and then that machine sequences it. So it's all extremely complicated machinery that's working behind the scenes to generate the genomes of these species. So we think of DNA as this kind of linear, chromosomes, A's, C's, T's, and G's, but inside of a nucleus, this is what it would really look like. It's all bundled up, kind of a hairball. And each of these different colors is a different chromosome. And the DNA is inside the nucleus interacting in different ways with different parts of the genome. And those interactions are really important. There's often reasons why particular bits of DNA, like the yellow bit here, is interacting with the green bit here. So the whole system that I just talked you through is we get the sample, we extract the DNA, we shear the DNA, we sequence in that PacBio, and then we add in these few other data types, which I'm not going to spend much time talking about today, but one of those that is called the high C sequencing. And instead of sequencing long molecules, it's actually trying to identify all these connections of how is the chromosome organized inside of the nucleus, so that you can find pieces of the genome that are nearer to each other and farther from each other. We also generate RNA sequencing data, which is the genes that are actually expressed in the organism. And then we put it all together, which we call assembly. So a reference genome, the high quality reference genomes we're trying to assemble, that just means put all the pieces together, are really important for the kind of biology we want to do. So I've added a tiny URL here for bgrabber, which allows you to go and see that exact specimen I just talked about, the data that we've generated so far for it. And this is kind of a quality control website where you can see the species that are in progress right now, but also the results for particular species. So this genome of this fly went into the sequencer, and just with this one technology, the PacBio, so we haven't added in this other information yet, you can see it's come out as, over here, it's about 300 million bases long is the estimate of how big the genome is. And most of the genome has come out in pieces that are 9 million bases long or larger. And there's only 104 pieces that this genome has come out in. And this is really a major leap forward for the project in this past year. This technology has really changed how we're able to sequence genomes. So far, I looked at that TollQC website where you can go and see the tiny URL bgrabber. And we have already started generating data for 59 species. One of the questions I know for Lyndall was around the hoverflies, and there are a lot of hoverflies in our project already because we are not only trying to do very big phylogenetic spans, but also take deep dives into some taxonomic groups. And so syrphids are one of the groups that we're doing a lot of sequencing in to try to get more species from multiple representatives from single families. So just a year into our project, we've already started generating data for about 1% of the fly species found in the UK, which is exciting. So what can you do with these reference genomes? I work primarily on Anopheles mosquitoes, and I'm not going to get into the details of these projects, but I'll give you a couple of highlights of the sorts of things that we can do. Genomes for maybe 20 different species in the genus, we've been able to look at the places in the genome which are common across all species and identify a more sophisticated way to do barcoding than the single molecular target of the mitochondrion, which is typically what DNA barcoding goes for. Anopheles is one of those groups of organisms where DNA barcoding doesn't work very well because there's a lot of interbreeding. So the mitochondrion can move between different species, and it's not a good marker for species level resolution. You really need to look at pieces of the genome to see what species it is. So we've created tools, because we have so many reference genomes, we can find these loci, these pieces of the genome where we can actually understand, if we go sequence these 60 parts of the genome, just small bits, so much more high throughput than the reference genome stuff I'm talking about, we can then identify what species a sample is. And we're going to be doing this at very large scale for African Anopheles mosquitoes over the coming years. As Erica mentioned, we've been working together with the Natural History Museum for a while to also try to understand what did their genomes look like before insecticides were in widespread usage. And in order to do that, again, you really need a reference genome. We can get tiny pieces of DNA, very degraded, 30 or 50 bases long, so 30 or 50 of those A's, C's, T's, or G's from these historic samples that Erica curates that are in the Natural History Museum, sometimes collected over 100 years ago. But if we don't have the right reference genome, we can't anchor those little pieces into a genomic context and understand anything about them. So reference genomes are really important also for looking back in time. And then another example is that we've sequenced the whole genome, so not using this kind of more sophisticated, really high quality DNA, but actually just taking short pieces and being able to align it to that reference genome and put all the pieces back together that way. We've looked at how insecticide resistance mutations move through populations. And the colors here, the red and blue, are kind of generally giving you two different species of Anopheles mosquitoes. And this is looking at a particular really important insecticide resistance gene. So this mutation has arisen and it confers insecticide resistance to DDTs and DDT and pyrethroids. And the interspersed red and blue that you can see over here on the right indicates that a resistance mutation has arisen in one species and then spread through interbreeding. I said Anopheles mosquitoes kind of don't obey species boundaries very well. So this allele has spread, this mutation has spread from one species into another repeatedly. That's what this back and forth red and blue means. So that brings me to the end of the sort of what do reference genomes do for us and why we're so excited about the Darwin Tree of Life project. And I also wanted to take the opportunity because this community of kind of fly fans is such an important community to talk to about another big project that we're going to be carrying out over the next 5 years. So BioScan, the Darwin Tree of Life project is a project that fits into this global endeavor, which is a kind of vision called the Earth Biogenome Project. So the Darwin Tree of Life project is really kind of the most far along and the most advanced in terms of all the different projects that are starting to pop up around the world to generate reference genomes for all 1.5 million described eukaryotic species that are out there. BioScan is another big global vision, which is instead of trying to get the whole genomes, it's really trying to change the way we do barcoding to not only target the species of interest, but also everything it's interacted with. And so if you imagine that you put a fly into a little tube and you run a barcoding approach on it, but you not only target the barcode that would let you know what the fly is, but also any plants that might be present or any fungi that might be present or any other parasitoids that might be present. You can actually get a marker for each of these different things and understand what is that fly infected with, what is that fly pollinating, what is that fly eating. And that is the premise of BioScan. So we will be kicking off BioScan in the UK using malaise traps in the coming year. So to sort of pilot this, I've set up a malaise trap in my garden for the past year, and you can see that this is the kind of what we're looking to do. We're looking to collect for about 24 hours in a trap, and then plate out all the specimens that come in that are killed into the ethanol in the trap. And then we basically, can do high quality imaging. This is not the high quality image that we would do, but gives you a sense of what these flies look like or these other species look like inside of wells of the plate. And then we basically are trying out right now different things, different liquids to put into these wells to make sure that we're preserving the specimens so that you can actually go back and morphologically study it, but we're also releasing its DNA, not only in such a way that we can barcode it, but also so that we can preserve it and use it for the Darwin Tree of Life project in coming years, when we know we will be needing to be tackling these much smaller organisms, which right now are not a major part of our project because it's hard to get enough of that high molecular weight DNA out. But we know that technology changes rapidly, and probably in a year or two, we won't need nearly as much DNA as we currently do need right now. So we release the DNA, and then we look at it. And this is that femtopulse that I mentioned at the beginning. This is how this is the output of that machine, and it tells you the fragment sizes of the DNA that's been released. So quite excitingly, we've been able to collect specimens. They've been sitting in ethanol in room temperature or outdoor temperature for 24 hours. And then we do this non-destructive extraction, and we look at the DNA that comes out. And we're getting the kind of lengths of molecules that we need for the Darwin Tree of Life project. So the plan is to set this up for the next 5 years across the country at 100 partner sites. And we'll be aiming to collect 10,000 flying insect specimens each month for the next 5 years. And so those will all be mailed to us at Sanger. We'll do the DNA barcoding, and we will image the insects. So these partners that get on board with this project will receive a malaise trap and the consumables that are needed. And at the Sentinel sites, which would be trapping, I think, a little bit more extensively. There's going to be about 10 of those across the country. They will also receive a dissection microscope to help identify and really get greater numbers of plating samples out. So it's going to be a partnership in which the people collecting will be basically spending their time and their effort to plate out specimens, and then we will sequence those and return back information on the species that we've identified there. So just to close up, I think it's really exciting times for insect genomics, and I'm really so keen to try to unite this bioscan project and these Earth Biogenome Project style endeavors like the Darwin Tree of Life to connect up so that we are future-proofing all this work that we need to do to barcode and to understand how species are changing over space and time, especially given climate change, and to make sure that all those efforts are future-proof so that we can actually generate high-quality reference genomes from those same specimens later on in the project. What next? Early next year, we're looking for to sort of pilot this in a little bit more of a large-scale effort than my garden, and we're going to ask these sorts of questions around, is this reasonable? Does it work? Are we capturing the kind of diversity we expect? What do we do with big specimens that don't fit into a plate? Do we have the right lysis buffer recipe? Do people who send their samples in want their specimens back? And how fast can we do this? We want to be able to do this relatively quickly so that partners are getting information back on the biodiversity at their sites relatively quickly. So I'm really open to suggestions, and I want this to be as valuable as possible to as many people working out there. So get in touch if you're interested in getting involved. And just to say that this is my last slide, just to say, in particular, I'd like to be focusing on some of the hyper-diverse fly families that are present in the UK. So with that, thank you very much. And I think I stopped sharing my screen now.
Thank you. I'm super excited about this project because it's just answering lots of questions that we all have. And apparently everyone else has. So I will start straight away with some questions. You deep diving into groups. This is kind of being, as someone else asked this as well, Barbara and John Ismay, and yes, in fact, so the acalyptrates they're very curious about. So propunculids and chlorophids and things like that, where there's some very closely related species. Will you be deep diving into those?
So this is why I need to connect with all of you, because I don't even know what that is.
Don't admit that. Don't admit that. Yes. Yeah, well, they are very diverse groups. And then again, we have so many problems in the UK with a lot of very, very closely related species. So this is.
So I mean, that is a big advantage of what we're trying to do with these, is that I think it will start to really give us a genomic perspective on how these things that are really difficult to tell apart, do they really look different at their genomic level? And does a barcode work or does a barcode not work? And can we actually, when we start to get enough of those skeleton reference genomes, can we start to build slightly more sophisticated barcoding methods that would allow us to then not have to sequence whole genomes all the time, but still tell us a lot more resolution on what species is what?
Yeah, really good question from Gabrielle Neve. How to distinguish whether what a specimen has been in contact with when alive versus one that it comes into contact with in the trap, in the Malaise trap?
Yeah, that's a really good question. And I'm hoping that the answer to that question is going to be that it's the numbers game. So the person who asked that question is correct. I think when a specimen is in the soup of the Malaise trap and there are hundreds of other specimens in there, there's going to be pollen released and it's just going to be floating around and it's going to end up on lots of different organisms. But when we see the same repeated interactions across many traps and across space and time, I think we'll have much more clear resolution about what is true interaction versus noise. So that's basically a signal to noise question. And I think it's the answer to that is that we just need a lot of numbers, a lot of specimens to find out what the signal is.
Okay. From Denise Warman, if I've pronounced your name right, sorry. Do species need to be collected into PCR clean tubes for the Darwin Tree of Life project?
Species need to be collected into barcoded tubes so that little orange cap tube, they need to be collected into those for this project. And I think that is Lindo mentioned it and I've mentioned it. I think right now where we are in the project, I think the best approach would be to get in direct touch if you want to collect, but also to think about whether going to Whiteham Woods might be an option if the specimens you want to collect are there, because then you can work with a really good setup where they have access to dry ice in -80s. And we know that we will generate high quality material out of the specimens that are collected. I think we still need a bit more time in the project before we're able to send out tubes. Unless if you want to send live specimens to Lyndall, great. But if you want to kind of collect into a tube that would go straight into sequencing, I think we need a little bit more time there.
Okay, and I'm going to ask one final slightly contentious question before we have our coffee break. As these technologies become cheaper and more accessible, are they going to replace traditional taxonomy and morphology? And that's from Oliver Lonsdale. That's one we all ask.
I don't think they will replace it, but I think they will have a really big impact on shaping our understanding. I don't see that traditional taxonomy is ever going to go away. I mean, we need to understand the biology of the organisms and the morphology of the organisms, and these need to also be curated, and we need to understand, there's a huge amount of effort that all of the museums will know intimately that I know much less well. So Erica, you should answer this question. That I think there's so much value in these collections that really the DNA doesn't give you on its own. You need to have that combination.
Yeah, see, now we can carry on working together. Correctly answered. Okay, Zoe, before we go to the break, do you want to say anything?